Hello. In this lecture we will study about bacterial cell wall. Bacterial cell wall is made up of a polysaccharide called as peptidoglycan. In this figure you can see a cross-section of bacterial cell. The left side of the figure shows cross-section of gram-positive bacterial cell. The outermost layer shown in purple is the cell wall, which contains peptidoglycan, underneath which is the plasma membrane. The figure on the right shows cross-section on gram-negative bacteria. The cell wall peptidoglycan is shown in purple color. Note that gram-negative bacteria contain an additional layer around cell wall. This outer membrane is shown in green color in the figure. We will now look at the structure of peptidoglycan. Peptidoglycan is a heteropolysaccharide. It is made up of two types of subunits. These subunits include N-acetylglucosamine and N-acetylmuramic acid. We have previously seen the structure of N-acetylmuramic acid when we talked about carbohydrate derivatives. This part of the molecule shown in orange color is N-acetylglucosamine. This is glucose on which the hydroxyl of carbon 2 is replaced by an amino group. And one of the hydrogens of amino group is substituted by an acetyl group. So the resulting molecule is N-acetylglucosamine. N-acetylglucosamine is in beta configuration. It is linked to N-acetylmuramic acid through beta 1 to 4 glycosidic bond. The other monosaccharide derivative shown here is N-acetylmuramic acid. N-acetylmuramic acid is actually a derivative of N-acetylglucosamine. The hydroxyl group of carbon 3 is linked to lactic acid molecule. The lactic acid part of the molecule is shown here. This lactic acid is in turn covalently attached to 4 amino acids. These amino acids include alanine, isoglutamate, lysine and D-alanine. This is called as tetrapeptide as it contains 4 amino acids. This is the basic repeating unit of peptidoglycan. In this next figure you can see the arrangement of this repeating unit to form cross-linked structure of peptidoglycan. In this representation you can see many chains of N-acetylglucosamine and acetylmuramic acid. N-acetylglucosamine is shown in orange color and acetylmuramic acid is represented in yellow color. Here you can also see that tetrapeptide shown by means of 4 green circles. The tetrapeptide is attached to acetylmuramic acid. The tetrapeptides of these chains are linked to each other by means of 5 glycines. These are shown in purple color. Since these 5 glycines cross-link the individual chains of peptidoglycan, so sometimes it is also called as pentaglycine bridge. An enzyme called transpeptidase catalyzes the attachment of these glycines and hence is important for the process of cross-linking of peptidoglycan chains. In this way, many chains of peptidoglycan are cross-linked with each other and form the bacterial cell wall. This is a representation from the other textbook. In the upper panel, you can see the fiber-like structures in the cell wall shown in blue color. Below, the same cross-linked structure of these disaccharides is shown. Penicillin is an antibiotic which inhibits bacterial cell wall synthesis. This figure shows the structure of penicillin. It contains 2 fused rings. The 5-cornered ring shown in red color is thiazolidine ring. It is fused to a 4-cornered ring called beta-lactam ring, which is shown in blue color. This CON bond is important for the activity of this antibiotic. Penicillin covalently binds to the enzyme transpeptidase. Transpeptidase enzyme is needed for the cross-linking of peptidoglycan through pentaglycine bridge. So when transpeptidase enzyme is inhibited, cross-linking of bacterial cell wall cannot take place. This results in fragile cell wall fibers which are unable to assemble properly and hence disintegrate. So these bacterial cells are unable to survive. However, some bacteria have overcome this. They contain an enzyme called as penicillinase. This enzyme is also called as beta-lactamase. As it breaks down the beta-lactam ring of penicillin. When the beta-lactam ring is disorganized, the antibiotic is no longer effective. A solution to this problem is to administer penicillin along with an inhibitor of beta-lactamase enzyme. So that if bacteria have acquired resistance against penicillin through beta-lactamase, penicillin would still act against them. One such inhibitor of beta-lactamase is called sulbactam. Its structure shown here. Augmentin, for example, is a common antibiotic used for many bacterial infections. It contains amoxicillin and an inhibitor of beta-lactamase enzyme and hence makes bacteria susceptible to the antibiotic.